mRNA vaccines contain instructions for your cells that tell them to build a part of a virus in large volumes.
So where in the body does this liquid travel? Well, it's injected in the circulatory system. The scientific term for the movement of any drug inside the system is called pharmacokinetics.
Starting with how the drug gets into the bloodstream in absorption, how it is distributed throughout the body in distribution, and how it is cleared by the body either through metabolism, through excretion, or by a combination of both.
Given this mRNA vaccine is going to change cells, you'd think it would be very important to the TGA approval as to where in the body it's going to end up.
TGA is the independent regulator who's responsible for overseeing the safety, quality and performance standards of vaccines. This includes COVID-19 vaccines.
Pharmacokinetics is generic to all drugs and vaccines, so therefore we can assume that they've addressed it. Well, they do mention, of course, pharmacokinetic, but only to say that there is a lack of data for the antigen provided by Pfizer.
And the TGA will not cut corners.
Well, one could argue that's not correct, because where this liquid ends up determines which cells are going to have their mRNA changed. So a couple of questions in regards to this liquid, that there's no data on where it ends up in the body, but it's in the blood, so pretty much anywhere. How many cells is the mRNA going to change? And very importantly, which cells are going to be caused to make this spike protein? Because if Pfizer hasn't told our regulator, the TGA, the pharmacokinetic data on this thing, if they do know, they're not releasing it anywhere.
Your immune system kicks into action and starts learning how to attack these intruders. Your immune system has learned how to attack the spikes of the coronavirus. It destroys all the spikes and even breaks down the mRNA vaccine itself.
Well, look, he's been a bit vague, but if it just destroyed the proteins, then obviously the mRNA would still produce more proteins. He says there that it actually destroys the mRNA. Well, the only way it can do that is to get inside the cell. Once it's inside the cell, is it going to like selectively just pick out that mRNA and destroy it? There's no more proteins. How's it going to identify the mRNA? I'll get to that in a second. Within a cell. I mean, it's just going to destroy the cell. It's the only way it can do it. Which, of course, we're now venturing into autoimmune diseases where the antibodies attack the body's own cells, which appears what's going to happen. Well, the TGA, true to form in not cutting any corners, also noted that in approving this thing provisionally, there is a lack of studies investigating the potential for autoimmune disease. So there's no pharmacokinetic data telling you where in the body and which cells are going to be producing this spike protein. There's also no data on whether it's going to constitute an autoimmune disease when the immune system responds to that protein being there. The autoimmune potential for disease set aside, if the cells are making the spike protein, what proteins are they not making, and what is the effect on the functioning of the body? Because they're producing different proteins now. Well, gene expression is a process by which the instructions in our DNA are converted into a functional product, such as a protein, so it performs a function. And gene expression is a tightly regulated process that allows the cells to respond to its changing environment. Now, the fact that it's tightly regulated means that everything our cell does through gene expression has to be precise. Now, this here is really important. So there's two key steps in making a protein. One is transcription, when the DNA in the gene is copied to produce an RNA transcript called messenger RNA, which is what this Pfizer nonsense is. So the second stage after transcription is translation. It occurs after the messenger RNA has carried the transcribed message from the DNA, protein-making factories in the cell called ribosomes. So there's the ribosomes in the cytoplasm, and they're going to eventually produce the protein. So here's a diagram of how a cell normally functions, and information from the DNA, as we saw through transcription, and that produces the RNA that then produces the proteins that are functional. So how does this Pfizer concoction enter a cell, intercept that process, and then insert the different RNA to then produce this supposed spike protein, that representative of the virus? And does it have to cut off the connection between the DNA and the RNA parts of the cell? Like, look how complicated a cell is. Like, is it affecting only the ribosomes, or is it going after some of these or changing some of the other characteristics of the cell? So the information from the media and from the TGA that they publicly release in their ads is very scant and doesn't go into any detail. So when you just even do a very slight dive into it, and that's all I've done in this video, you could, first of all, you could say that the TGA is irresponsible in allowing provisional approval use on the Australian public for something they don't have any autoimmune data on, or where in the body and which cells are going to be affected. So these unanswered questions that aren't answered out in the media, that is enough. And they're unanswered because there either is no data or they're withholding the data. That's enough to say, okay, it's over, I'm not taking this thing. Because theoretically, you got blood in your brain. This thing could land in your brain, enter cells in your brain cells, and start producing spike proteins, which everything's tightly regulated, remember, so it functions correctly. I mean, that could have impacts on your vital organs, on anything. Now, add to that this character has signed off on exemptions for the legislative requirements on labels for this thing, which would ordinarily limit and determine what is in those vials. Means that at the point of entry, sure, even they're already very dangerous concoction into your system, doing things to your body that shouldn't ordinarily be happening. Well, they could then add anything else and any other changes they want to make to your body. So I just want to finish off with pharmacokinetics includes clearance and half-life, of which there is no data, and the fact that there is no long-term safety data. The TGA has come out and said we've only approved this thing for short-term safety. So what if it's the case that your system can't clear the mRNA from this concoction, which comes back to even in this video where the guy says that the immune system's going to destroy the RNA. Well, that's part of this pharmacokinetics data, which how could he know that? Because if Pfizer, like I said, hasn't released it to any regulator, how can the media, because the media have also said, it's only in your system temporarily. Well, they can't know that because the pharmacokinetics data hasn't been released by Pfizer. So, all right, that's it for this one.